Okay, in this last section, we're going to hit some of the odds and ends associated with the vessels and the cardiovascular system. So the first thing we're going to talk about is blood pressure. Keep in mind when we talk about blood pressure, one of the things that we've already discussed is the effect of blood volume on blood pressure. And you can actually see this being re-emphasized when we talk about the renal factors. Remember that the kidneys are responsible for filtration of the blood, for the amount of volume of urine, in other words, how much water we actually remove from our body and void, and the kidneys then determine the total water volume we keep. Remember from one of the previous systems, we talked about ADH, or antidiuretic hormone, and ADH's role in the ability of kidneys, or how it causes kidneys to control the amount of water we retain or the water we remove. So that's the most basic control, is we simply look at the amount of blood we have. Now, other factors include neural factors. In other words, we looked at how the nervous system can control the heartbeat. Heart beating and the amount of blood that I am moving impacts my blood pressure. Temperature is going to impact blood pressure, whether you have vasodilation or vasoconstriction. And then different chemicals can have an impact on increasing or decreasing the blood pressure. And then diet. Here, diet kind of covers a lot of different things. This would be everything from cholesterol, which has kind of a clogging effect on the arteries, especially if it's the wrong kind, not the good kind. Salt intake, because the amount of salt that you have in your body directly affects the kidneys and how much water is removed from the urine. Different things like that. Now, the book has this really nice figure, 11-19, that walks you through some of the different homeostatic mechanisms controlling blood pressure. This is something that's definitely worth you recognizing. In other words, notice how vasoconstriction and dilation has an overall effect on the blood pressure. Everything here we're looking at negative feedback on again. So recognize that if I have a stimulus of cold, I see vasoconstriction. This is going to have an overall effect of increasing the blood pressure because you're having to force the blood through smaller openings, whereas vasodilation will have the opposite effect. Now, one thing that you want to keep in mind is we try to create an average, but it's not like an average. It's not like we take all of the people and try to average what their blood pressure should be. What we come up with, or what most of the medical professionals come up with, is an acceptable range. Now, this has changed, actually. Normal is no longer 140 to 110. It's actually a little less. I think it's going to be actually 130, or I'm sorry, this is the systolic. 130 is the ideal. 130 might even be the peak now. The diastolic, 75 to 80 is about right. One of the things to keep in mind also, if I hit 100 on the diastolic range, that's actually more dangerous or equally dangerous than as hitting 200 on the systolic range. Those numbers are way outside of normal and are considered serious enough that you should actually try to get medical attention if they hit that range. Hypotension, too low blood pressure. Hypertension, which is what we hear a lot more about, is high blood pressure. Hypertension, again, affected by all of those things we looked at in terms of diet, chemicals, temperature, different neural factors, etc. Now, the next thing we're going to talk about are basically the function of the capillary beds in the body. We have to have, every cell in our body has to get adequate oxygen. We've already established that all of the oxygen in our body is carried on our red blood cells. Red blood cells are tiny, but a single red blood cell can carry up to 1 trillion molecules of oxygen. How we then get the blood that's in arteries and veins to the cells, which we know are really, really small, is via capillary beds. Capillaries basically force the blood to come in single file. So literally there is just enough space, or the vessel is just of the size that the red blood cells literally almost line up single file and come through it. Now, how we control capillary flow, and this ties back into what we've talked about before in terms of muscle contraction causing an initial drop in blood pressure because suddenly we have blood flowing into capillary beds that weren't as active as they are now, and that's these things. And what we have is we have different sphincter muscles, which are just little pieces of smooth muscle that will contract or not, and so they contract or relax, and that allows blood to flow through different capillaries based on need. If I have an area that doesn't get or doesn't need a lot of blood flow, I'm going to only sporadically allow blood to flow through an area, because it doesn't need to flow through all the time. It doesn't need to flow there all the time. It needs to flow enough that my cells stay alive and stay healthy, but it doesn't have to occur constantly. So what we see then when we do have blood flowing to particular areas is we have what's known as capillary exchange. We see all the good stuff come in, we see all the bad stuff leave. And the bad stuff is what's basically associated with metabolic waste, carbon dioxide, uric acid, all of that stuff. Now, at the capillary beds, we have two primary driving forces. We have basic diffusion, so concentration gradients. We also create what's known as osmotic pressure. Remember that the blood has proteins specifically for the purpose of maintaining an osmotic balance. We want to maintain a certain amount of fluid in the vessel that doesn't flow out. Well, the very presence of that pressure within the vessel also helps to move some substances in and out of the cells. And so we see this movement, okay, through the pores of different substances. We see, remember that when we talked about leukocytes have the ability to travel through and between cells and the gaps in the cells. And we also have the pressure from within the capillary that helps to move forces some of the nutrients to exchange and some of the gas exchange to occur. Okay, the last thing is a quick reminder about how the heart developed. Remember that we see by the 4th week we have basically a pumping heart that is self-regulated, and then becomes the 4-chambered by 7 weeks. And again, this is just a quick review from the video we had on fetal development.